but we don't know. We'll have to see.
Yeah. It has some advantages for students.
Yeah, our main worry now is just watching the death rate, and we know cases are going to rise very quickly. We're just hoping that the booster jabs have been given out and that the death rate stays below 500 a day. That would be good, but we don't know.
Yeah, but how about the admission to hospital?
Yeah, at the moment it's still very, very flat and level. So I think the UK at the moment has about 100 deaths a day, which is still bad, but it's relatively low compared to what it was. And that's going to increase 2 weeks after Omicron starts, then you get hospitalization and death rate going up. So only time will tell.
Okay, sorry. Akil, excuse me, maybe you can mute your microphone, Akil. Hello, Akil. Okay. Sorry. Okay, it's 8:30 now, UK time. I think I will start. Okay. Good afternoon, everyone, and good morning for Dr. Philip Drake. I would like to say thank you for joining today's guest lecture of DECOTA video conferencing with Universitas Airlangga, entitled COVID testing with lateral flow antigen test and PCR-based DNA test by Dr. Philip Drake from Faculty of Life Science, University of Bradford, UK. Thank you, Dr. Drake. It is a pleasure to have you here with us for sharing about the COVID testing knowledge. And this DECOTA is part of molecular cell biology subject, nanotechnology engineering study program, Faculty of Advanced Technology and Multidisciplinary. Before we begin, I would like to remind second-year students to make a page of resume of today's talks. First thing first, I would like to introduce a short CV of Dr. Drake. So Dr. Drake is a lecturer in chemistry department, University of Bradford, and now he is a hold a leader position for MSc program. Dr. Drake was graduated from University of York with a Bachelor of Science honors in chemistry in 1995 and continued his PhD in polymer chemistry in 1999. And he is currently a lecturer in chemistry at University of Bradford since 2014. And he has some experience such as working at Defense Evaluation and Research Agency, part of the Ministry of Defense in the UK, and in 2002 up until 2014, he was working in the Industrial Technology Research Institute, Ministry of Economic Affairs, Taiwan, where he began to focus on polymer chemistry and material science for biomedical application, where led him to have 2 patents, and I guess we'll have many more, I hope. And he has several papers in reputable journals and focuses on the project of polymer chemistry, nanoparticles, chemical sensor, and many more. And Dr. Drake will present 45 minutes, and there will be 15 minutes for Q&A. Okay, without further ado, I will offer the spot to Dr. Philip Drake. Please take your time.
Okay, thank you for that introduction. It's Christmas here. This is the last teaching day in the UK for our university, so I've got my reindeer antlers on for now. I'm going to take those off now. I won't talk with those on, hopefully, if I can take them off. Okay, that's not working. I've changed to something else. Okay, I have to get non-background. There we go, antlers are gone. Right, so let me start my talk. Thank you for the introduction. Today, I'm going to be talking about COVID testing and the use of lateral flow antigen tests, how they work, and PCR-based DNA tests and how they work. This is a talk I gave at the start of the COVID outbreak for the National Science Festival in the UK, which was part of the Science Museum's Festivals of Science. So this talk is aimed at people of various different backgrounds, just to understand what is happening with COVID testing and how they work. I'll also introduce myself and the research I do. So let's get going. So this is me. I'm just I've got lots of little icons open. I'm trying to remove all those so I can see my screen. Get rid of that one, that's okay. So this is me. I'm on Twitter. This is my Twitter handle, @materialchemist. So feel free to follow me on Twitter if you like. My research is in material chemistry and nanoparticles. And I look at point of care technologies, in particular SIRS-based technologies where we're using lasers to detect pathogens, detect bacteria, detect viruses. I also work in the area of cancer therapy where I'm using nanoparticles again and magnetic field hypothermia to try to heat up cancer and to kill cancer. So the SIRS-based pathogen detection is applicable to this talk, and if I have time at the end, I might briefly mention it, but that's a whole new talk I could give in the future. Equally, the use of magnetic nanoparticles for cancer therapy is another talk which I can give in future. But for right now, I'm going to focus on lateral flow tests. This is my email address, p.drake@bradford.ac.uk. So feel free to email me any questions you have. I'm always happy to talk about work and to answer questions from students. So do feel free to contact me. So today I'm talking about bioassays, what they are and what they do. I spent a lot of time in Taiwan looking at unmet medical needs, how I can apply chemistry to improve medical detection of biomarkers. Bioassays are very common. You can buy them over the counter in the UK. Things like pregnancy tests you can do. They're based on a lateral flow test. They're very similar to the COVID tests, lateral flow tests we can do. People should be very familiar with those, but there's also things like breath analyzers where the police use them to look at the breath of drivers to see if they've been drinking alcohol. So the breath analyzer is another example of a bioassay. So a bioassay is simply where we're using biological methods or we're detecting biological samples, could be breath, could be blood, could be urine, and we're looking for chemicals in those media to see if we can relate those to disease, to conditions, to other things. They're also used in forensic science. So one of my lectures, I lecture on detection of drugs and pharmaceutical drugs. So I do a lot of work with forensic science, looking at urine, saliva, and hair testing. Again, those are all samples of bioassays where we can look in the urine, we can look for drugs, we can look for biomarkers. We can detect the health of someone's body by looking at the urine. We can also do the same things with saliva and blood. Hair is a very interesting method, especially with drug analysis. We can detect drugs in the hairs of people, even though they haven't used them for 1 or 2 years, we can still detect evidence of drug use in the hair samples. So hair is quite a nice little sampler. And also more advanced methods like DNA fingerprinting and DNA testing. This again goes back to DNA sequencing and how we can use DNA technology to detect viruses. But it's all based on PCR process, which I'm going to cover today. We then have medical tests in hospitals where they're looking directly in the blood. They're looking for things like cancer markers, markers for heart attacks, markers for strokes. More commonly, there's a diabetes type detectors where you're looking at glucose in the blood, how we can detect that. These could be based on lateral flow systems. It could be based on PCR. It could also be based on electronics and electrochemistry. So there's lots of advanced methods to looking at bioassays. And then finally, we get to detecting bacteria, detecting viruses. So this is where my work fits in really, is how we can improve the detection of bacteria using nanoparticles and advanced chemistry. So before I get into the talk, we need to have a little understanding of what a virus is. So again, this talk is aimed at the general population. So I'm not sure what background you have with viruses. So forgive me if I'm going to too basic a detail, but it's interesting to know what we're looking at. So a virus is a very small structure. Some people say they're not living systems. They can replicate, they can reproduce. They're very, very advanced little bits of technology, and they're a really good example of what nanotechnology could in future achieve. So this is the common flu virus, I believe, the influenza virus. It has a round structure. It has these proteins on the surface, these spikes. Inside, it has its RNA wrapped up in a little coil. RNA and DNA are the same kind of things. So RNA is a slightly different version of DNA. It's ribonucleic acid as opposed to deoxyribonucleic acid. And we can use enzymes in the body or enzymes outside the body to convert RNA to the same DNA sequence. So they're considered to be similar, and they can alternate between the two. There's a very interesting story of RNA and DNA and how it fits into evolution. But again, that's another lecture. They don't have to be spherical shapes. They could be fancy shapes. They can be these hexagon shapes. This is the polyhedral adenovirus, slightly different shape and structure. They could be helicals. This is the tobacco mosaic virus. It's a very different shape again. They all have the same basic function. This is one of my favorite viruses. This is the bacteriophage virus. It always reminds me of mosquitoes, this virus. It acts exactly like you'd expect it to. It comes down, it finds a cell of target, it attaches to the cell, and then it injects the inside. So it bends down. The RNA from inside the virus is then injected through a channel into the cell of a target. So this is the bacteriophage. They don't have to be round, it can be any shape, but they're all amazing little things. When a virus infects a cell, the proteins on the surface recognize receptors on the surface of a cell. They bind to those surfaces and they get pulled into the cell. Once they're inside the cell, the virus then opens up and releases its RNA. The RNA then reprograms the cell to start making more virus proteins. So once the RNA is removed into the cell stops becoming its standard cell. It could be a lung cell or a heart cell or a liver cell. Instead, it starts to produce more virus proteins. These virus proteins build up. They're expressed on the surface. So now, rather than being the cell expressing its own proteins, the cell now starts to express virus proteins. Inside the cell, the RNA is replicated, packed up into a little package. It goes to the edge, it bubbles out and forms a new virus. It could be thousands of more viruses coming out from the cell. These viruses then go on to infect more cells. So that's how a virus works. That's how it infects a cell. And that's why COVID-19 is so dangerous. When it starts to infect the lung cells, the cells within the lungs which help us to breathe stop working. They can't transfer the oxygen and you start to die from suffocation. So how big is a virus? I'm all about nanotechnology. I work in the nano world, which is a very small world. But a lot of people find it hard to picture how small nano is. A virus is around 100 nanometers in diameter. The COVID-19 virus is about 100 nanometers in diameter. This is a picture of human hair. I don't know whose hair it is. This is the skin. This is some hair sticking out of the skin. I got this from Google. It's just a random picture. If we zoom in on the skin, on the hair follicle, we can see the hair shape. This hair, a single piece of hair, is around 100 micrometers across from side to side. This scale bar tells you that's about 10 micrometers. The whole distance across the hair is around 100 micrometers. Red blood cells. You should all be familiar what a red blood cell looks like. That's a picture of a red blood cell. We know that red blood cells are around 7 to 8 micrometers across in size. So this now is a red blood cell drawn to the same size as a human hair. Red blood cells, about 7,500 nanometers, 7.5 micrometers. 7.5 times 10 to the minus 6 meters. This little dot here is the size of the COVID-19 virus, 100 nanometers virus particle compared to a red blood cell. So that's how small a virus is. It's much smaller than a red blood cell. They're very hard to find. We know from modeling and from pictures that this is the shape and structure of the COVID-19 virus. This is an atom level model of what we know the virus looks like. These proteins on the surface are the spike proteins, the envelope proteins. These are the proteins which bind to the cell surface that recognize certain cells and find their way into the cells. And it's these spike proteins that we have our antibodies designed to detect. This is a model. This is a real picture, TEM picture of some coronavirus particles. They're called coronaviruses because you see this corona shape around the top of the virus. We have a round spherical virus with this corona on the surface. This corona is produced by the spike proteins. Inside the virus, here we can see a virus that's been broken in half. Inside this virus, we can see the RNA wrapped up and coiled around. So this kind of model, the atom level model, comes from these photographs. The TEM, the researchers find lots of different virus particles. We take lots of pictures of them on a TEM. We get all the different shapes. We can then put those into a computer, and the computer models all the different possible angles taken from the images. So from 1,000 images, you get the picture of a full spherical protein virus. And we can also see the inside. We can model that. So we know that's 100 nanometers in size. That's what it looks like. Inside the virus is the RNA. So this is now the RNA. It's wrapped up in a very tight coil, very carefully packaged inside the virus. And we can see that here on this structure. So this is the model. This is the real image, and that's how well they fit together. So we know we have these spike proteins. The spikes on the surface of the virus give a look of a corona surrounding the viron when viewed by TEM. In this view, the protein particles E, S, and M located on the outer surface of the particles have been labeled. So this is your E proteins, your S proteins, your spike proteins, and your M proteins. Novel coronavirus named acute severe respiratory syndrome coronavirus 2 was identified in 2019. It's since gone around the world. It's mutated a few times. We've had the Delta version. Now we've got the Omicron version, where the sequence of these spike proteins changes slightly. And this is what gives rise to COVID-19 infections. So this is a real transition electron microscope image of the isolate from the first US case of COVID-19. So this is a cell, human cell, that's been destroyed by the viruses. The viruses have been released. These are the viruses plumping out from the cell on the surface. This is from the real images from the first COVID-19 case in America. When a virus first enters the world, we don't know what it is or what it looks like. All you can do is look through a TEM at images of cells and look for evidence of viruses. When you find these viruses, they can then be extracted from the sample. We can do some DNA sequencing, some RNA sequencing. We can find the genome of the virus. We can then start to model the virus, model the shape, and work out how to detect it. But the first step is always trying to find the virus, and that is really difficult because they're so small. We can look at thousands and thousands of pictures of different cells and not find any viruses. But eventually they get found, and we can start to detect them. So this is the model of COVID-19. If we turn that to a black and white image, you can see how the model fits with the actual image you see in real life. So what's the timeline of infection? When COVID infects the body, what starts to happen? Day 0 is when you start to feel ill. You're actually infected a few days before that. So maybe up to 5 days before that, the virus enters the body. It enters the body and starts to replicate. It replicates and it grows. The virus concentration in the airways increases over time until eventually it starts to make you feel ill. Then you have symptoms, you know you're ill. At this point, we can start to detect it. We can do nucleic acid tests. These tests for PCR sequences, they test for the virus itself. So when you have a high virus load, you can do a PCR test to detect the virus. At this point, you've got no antibodies. Your body starts to detect. A few days after infection, your body realizes something's happening. It starts to produce antibodies. But it takes a long time for the body to design its antibodies, to produce them and start to produce enough to kill the virus. So antibody production begins, but it's very low. After about a week, your antibody production is going fairly well. You're producing more. Once your antibody production goes up, your body starts to kill off the virus particles start to go down and disappear over time. At the same time, your antibody levels increase and go up. So at this point, about a week after infection, you can do an antibody test and you should be able to detect antibodies in the blood of people infected. So this is the antibody lateral flow tests. There's always a bit in between where the virus particles are going down, your antibodies aren't very high. So at this point, a few days, it's quite hard to detect if you're infected or not. So that's the timeline of infection. The particle goes up, your antibody starts to develop and increase. So the first way of detecting the virus is when you have a high virus load. It's nucleic acid tests, the PCR-based tests. So they're called different things. It could be called a NAT test, which is nucleic acid amplification test, more commonly known as a PCR test, polymerase chain reaction test. So the PCR tests were invented and developed by Karen Mullis in 1983. She got the Nobel Prize in Chemistry for doing that work. It's all based on DNA detection. So you have a target DNA, a known sequence, double-stranded DNA. In this case, we have a red strand and a blue strand. But DNA is alpha helix, it's 2 strands of DNA intertwined. It's easier to look at it as a red and blue sequence. We also need primers. These are little sequences which we know. We know the sequence of the DNA because we found the virus, we've extracted the genome, we know the sequence we're looking for. If you know the sequence of DNA, you can design a primer which will bind to a certain area of it. It's usually about 20 nucleic acids long. So you're looking for target DNA, double-stranded DNA. If you heat DNA up, the 2 twines alpha coil will start to uncoil itself and then become separate. So you go from a double-stranded DNA to single-stranded DNA simply by heating. It's called denaturing. So we heat the DNA up and it denatures and forms 2 single strands, the red strand and the blue strand. We then add in our primer. So our red primer is designed to attach to the blue strand. Our blue primer will recognize the red strand and they'll bind to it. So by cooling this down, we anneal the primers. The primers will attach to the DNA sequence. There's a low concentration of DNA, a high concentration of primers. So the primers bind first. Once the primers bind, the DNA itself can't bind to each other because the primer's blocking it. So that's the primer binding. We then go through extension. So we use polymerase, an enzyme which will detect the double-stranded section. It then reads along and rebuilds the missing DNA. So it binds to your primer section and then rebuilds the missing part. It's called extension. And that's simply done by heating the mixture up again. So extension occurs, at the end of extension, you've got 2 new strands of DNA, identical strands, the red one and the blue one, double-stranded DNA. So you go from a single strand of DNA to 2 strands of DNA in one cycle. And the cycle is simply cooling, heating, cooling, heating, giving you a reaction. So that's one PCR cycle. You start off with 1 strand of DNA. After 1 cycle, which takes about a minute, you have 2 strands of DNA. You've doubled your number of DNA. After 2 cycles, you double that again to 4 strands. After 3 cycles, you get to 8 strands, and you keep doubling all the way through. After 4 cycles, you get 16 strands. After 10 cycles, you have about 1,000 strands of DNA. After 20 cycles, in my lecture I give to my MSc students, I ask them to guess how much they'll have after 20 cycles. Have a quick think about it. How much DNA would you have after 20 cycles? Because we don't have much time to go through it, I'm going to give you the answer. It's around 1 million strands of DNA. So after 20 cycles, you've gone from 1 piece of DNA to over 1 million strands of DNA. And each cycle takes a minute. So that's about a 20-minute test. We use a dye called cybergreen. So cybergreen dye is a fluorescent dye. And it's a special dye because it will only fluoresce when it's inside double-stranded DNA. If it's outside the DNA, it won't give any fluorescence. So it only fluoresces inside the DNA. So what this means is at the start of the reaction, you've got your primers bound on, you've got a little bit of fluorescence from the primers, but nothing else. The rest of the cybergreen isn't fluorescing. As the reaction proceeds, you get lots more DNA, the fluorescence increases. So cybergreen is a fluorescent dye that only emits light when it's inside double-stranded DNA. It's the environment of the DNA, the hydrophobic environment, that causes it to fluoresce. So in a negative test, there's no viruses in the blood. There's no RNA present. There's no DNA present. So when you go through your cycles, you get no fluorescence. So if you plot a graph of fluorescence intensity against cycle number, what you see is a straight line fit, nothing's happening, no fluorescence is increasing. That's a negative test. In a positive test, the virus RNA is there, it's converted to DNA, the concentration of the DNA increases every cycle exponentially. So as you go through the cycles, the fluorescence intensity increases as you go. So you start off with low fluorescence and you get this S-shaped curve where suddenly you go from low, fluorescence increases exponentially and then suddenly levels off again. So that is a sign of a positive test, this sudden increase in fluorescence. We then have an arbitrary point. We put in a threshold level. So we think, okay, once the fluorescence goes above a set level about here, how many cycles did it take? So we set in a threshold level, and then we can get a CT value, the cycle number when it went over the threshold level. And it's this CT value which they can relate to how much virus is present. So this is one of the problems with PCR testing is, where do you set the threshold level? It's debatable. Some people set it high, some people set it low. So it could be set very low, it could be set very high. Equally, where do you put a CT value as being a positive test? Is it after 20 cycles? Is it after 30 cycles? Is it after 10 cycles? So normally for a virus, we know how the virus infects the body. We know how the virus will adapt and change. So we set the CT value when we know the person is infectious. If they're not infectious, it doesn't matter. They won't pass the virus on. For COVID, we don't know where that CT value is. We don't know where the threshold value is. So at the start of the pandemic, we set those very low. So everyone was testing positive. Even if you got one virus in your body, a dead virus, a piece of DNA, you could still detect it, and that could still give you a positive test, even though you're not really infectious. So as time evolves, we get to know more about the CT values and the threshold values. The whole point of PCR systems, especially the work I do in the nanoparticle PCR, is to quantify how much virus you have based on the CT value. So if you start off with a high concentration of DNA, high concentration of virus, you get a very low CT value. So first curve, your CT value is something around 5. As concentration of the virus goes down, the DNA level goes down, your CT value goes up. So you can plot a little straight line graph of CT value against virus concentration. And again, that's where this is data from my actual research. This is some PCR-based nanoparticle data, which I might cover at the end of the talk if I've got time, or maybe in the future I'll give another talk about that. So that's PCR-based. You take a swab, it could be blood or it could be saliva. From the swab, if you've got virus particles, you extract the virus DNA. The virus DNA is then converted to virus RNA is then converted to DNA. You go through reverse transcription. You then add in your dyes. You do a PCR-based test. If fluorescence increases, it's a positive test. So you have a positive test or a negative test. And that's just how the PCR systems work. I got these images from Compound Interest. It's a very good Twitter account, so do follow Compound Interest and look at those details. They have some very nice things about Christmas and chemistry. So it's a nice site to look at. That's the PCR. The other kind of test we do of a lateral flow test looking for antibodies. So how do antibody tests work? So the COVID-19 antibody serological test, it's a blood test. It doesn't have to be a blood test. It could also be a saliva test. It's designed to detect antibodies, immunoglobulins, IgG and IgM, against the coronavirus that caused the disease COVID-19. The antibodies produced by the immune system in response to infection or equally from vaccination. So a little bit about antibodies. Antibodies are a Y-shaped molecule. They're fairly big, 150 kilodaltons. For me as a chemist, that's very big. For a biologist, it's not too big. The two strands at the top are called the FAB strands. These two are exactly the same. The third strand down here is the FC strand. That's different, has a different structure. It's the tips of these two strands where the antibody will detect its target compound. They have a variable region. This variable region can change. Every antibody will have a different sequence here, a different shape, and it's the shape that binds the target. So via the vaccinations we have, they're designed to detect a spike protein. When it detects a spike protein, it will stick to the tips of the virus here. The FC is different. We can use the FC region to attach antibodies to surfaces. So if you take an antigen, COVID-19 virus, it's got the spike proteins on, our antibodies will detect those spike proteins, they'll stick to them and bind to them. So they bind around the virus. In your body, when this happens, your body now recognizes this virus as something it needs to attack, and T cells and B cells come out in your immune system and start to destroy the virus. If you have COVID-19, you'll have antibodies in your blood. That's the IgG, the antibody to COVID-19. These antibodies bind to proteins on the virus surface. So these tips will recognize the spike proteins on COVID-19. When they come across it, they'll just stick to the surface and tell the body to destroy the virus. We can test for these antibodies using a lateral flow system. So lateral flow immunoassays are a very simple way for anyone to do very quick tests. For this, we need several special components. So there's lots of different things within a lateral flow system. We need some chicken antibody. Sounds a bit funny. We have chicken antibody that will recognize human IgG. So this is chicken IgG, which will bind to the FC region of any human IgG. We can make that in the lab. We can produce it in the factory. It's fairly easy to make. We also need a label. They're usually gold nanoparticles. So a label is just something you can see. The gold nanoparticle has a very good red color, which makes it very easy to see. So you need some gold nanoparticles. You also need an antibody that recognizes chicken IgG. So this is another antibody, and this antibody will recognize chicken IgG. We also then need some virus protein. We know the sequence of the proteins. We can start to manufacture them. We can produce this quite quickly in an industrial setting quite well. So we need some virus protein. If you've got all these components, you can make a lateral flow test. You take a strip of paper. The capture antibody and the virus proteins are stuck on the surface. So this is your strip. You take your anti-chicken IgG and you place it on the strip. You attach it to the surface. It's now chemically bonded to the paper. It can't move. It's stuck on the surface. And you stick the FC region. So the two FAB regions are sticking up. That's your capture antibody. You take some virus protein dye. So that's the virus protein sticks on there. And you stick that to the surface too. The reagent stored at the start of the strip is labeled with a dye molecule, often a gold nanoparticle. So we have some chicken anti-human IgG with a gold nanoparticle attached. And that sits on the top. It's not attached. These two are attached to the surface. This one just floats on the surface. Place a drop of blood on the strip. So you put your blood at the beginning here. The blood contains antibodies to IgG. It'll give a positive test. So if that has the antibodies in, it'll be positive. That's the COVID-19 antibody. The blood flows down the strip. It washes these reagents down. They all interact. Your chicken IgG at the start will stick here. That's your capture body. That's your control line. That should always give you a red line. If you've got antibodies, they will bind to the protein on the surface. If that binds, your chicken IgG will also bind to that one. So that gives you a positive test. If your blood doesn't have any antibodies in it, you get a negative test. So this flows down. Your control line is still activated. You still get a red line, but your sample line, your test line is negative. Only one line will appear on the strip. So for a positive test, you get two lines, two nanoparticles bind, you get two lines on the strip. For negative, you get a single line. And that's what it looks like. So positive is when you have two lines. It could be a very faint line. If it's still there, it's still positive. A negative test is when you have just a control line and nothing else. If you get a test on the positive here and nothing on the control, that's an invalid test. That means that the strip has gone off, the antibodies aren't working, something's not working properly. It's invalid, you have to do the test again. So this is when we're detecting the antibody in the blood. We can also change the test round, and instead of taking for antibodies, we can actually just test for the COVID-19 protein. So in that case, we have a capture antibody control line. If you've got a protein in your saliva from the COVID, it will stick to the antibody. Your label will then stick to that, and you get two lines. So we can adapt the method not to look for antibodies, but to look for proteins, spike proteins from the COVID. And again, that's a positive test, that's a negative test. So that's how the lateral flow tests work. Again, it's a fairly simple process. You take a pinprick, you put the sample in, you give it a mix, you put a few drops onto the test strip, you wait for a bit, you see how many lines appear. It's exactly the same process as you do for a pregnancy test using from urine. You could also use saliva rather than blood. and you can look for proteins instead of antibodies. I've been doing these tests now pretty much 3 times a week since the summer. We have to do our tests all the time. If it tests positive, then we don't go to campus and don't give our lectures. We do our lectures online. If it tests negative, we can keep going to campus. So that's how they work, the lateral flow systems. This is just another graph to show you the different areas. So the antibodies will increase as you get infected. They increase in different parts. So a nasal swab will give you positive, slightly different times from a bronchial swab. After you've been infected, you start to develop your antibodies. This is where antibody production begins. You actually make 2 different kinds of antibodies. We call them IgG and IgM. So the first time you're infected with COVID-19, you produce something called IgM, and you produce that earlier than IgG. IgG is your normal antibody. After you've been infected and you recover, your IgM levels drop back down to 0, but your IgG stays fairly high, and that gives you immunity from the same virus again in the future. So IgM is another thing we can look at. This is IgG. IgM is a pentamer of IgG. So it looks like this. It's quite a large molecule. It's got 5 different bits of antibodies on. The structures are slightly different, but the main binding points are the same. This is IgM. We can add a 3rd line to the immunoassay strip if we wanted to and look for IgM as well. This can tell if a person had a recent infection. So by looking for IgM, you can get a bit more information. The same process, you have a test strip, you have your capture lines, your control lines, you have your IgG line, you also have an IgM line now as well, so you have more capture antibodies. If you do that, you get a more complex thing. A negative test when you just have your control line positive. You might have an IgM line only and a control line. Now this means you've been infected and it's very recent infection. You might have control and IgG only. Now this means you've been infected, but the chances are you've recovered. You had COVID maybe a month ago. Or you might get all 3, which means you're currently infected and you've been infected for about a week. So this is recent infection within a week. This is past infection, and this is ongoing infection, maybe 2 weeks old. So by looking at IgM levels, this is some real data, some real pictures of what it looked like. You can see the 3 lines. That's an IgM only positive, a negative test, IgG only test. That's IgG only as well, and that's IgM only. So looking for IgM would also be quite interesting. So IgM is good because the first time you're infected, you produce it and then it disappears down to 0 in the body, whereas IgG stays higher. This is our innate immunity that stays in our body. So once your body has learned to produce a virus, IgG, it keeps producing a lot quicker. And that is the end of my talk. Now, I've still got 10 minutes left, so I'm going to start talking about the lateral flow tests I did with nanoparticles and how we can improve these kind of systems. So thank you for listening, but I'm going to keep on going for another 10 minutes with some more details. So my work is looking at how we can improve PCR methods, how we can improve the detection method. And I like to use SIRS. It's a surface enhanced Raman spectroscopy where we're using lasers to look at samples. We can make nanoparticles that give a very strong SIRS signal. So this is a gold nanoparticle. If you shine a laser onto this, you get some scattered photons, which you can detect very easily. We'd actually get single molecule detection. So we can detect a single nanoparticle in solution. I combine this with magnetic nanoparticles. So these are magnetic iron oxide particles. To do this process, we have the primers. So back to the PCR, you have PCR primers. If I put the primers onto these nanoparticles, so my gold has the reverse primer, my magnetic particles have a forward primer. Then this is the target DNA we're looking to detect. This is the COVID-19 DNA sequence. If this is present, then it will bind to the primer. It'll bind to both primers. If it binds to the primers, then you get the 2 nanoparticles linking together. You get dimerization. So at this point, we have a SIRS active particle, we have magnetic particles, but they're not attached. Here we've got a SIRS active magnetic nanoparticle attached to each other through a DNA link. And that's the concept of improving the PCR system to use SIRS. If we go through the PCR process, what happens is the DNA sequence attaches to your magnetic nanoparticle on the primer. You then get extension occurring. So this DNA sequence on the magnetic particle is then extended to the full DNA sequence. Once that extension has occurred, the primers on the gold nanoparticle can attach to this point. So they recognize this bit of the strand and they attach to that, and you get dimers occurring. You then get extension from another primer on the magnetic particle, and that extends. So what you end up with is a large complex structure of alternating nanoparticles, gold and magnetic. This is what the magnetic nanoparticles look like. We have an iron oxide core, magnetite core, with a glass shell. This is what I was hoping I would make. The glass shell is encoded with amine groups. So this is a real TM image of the particles we make. We can do some analysis on the surface here. So we look at this point here, using electron beam going down through the surface. We can look at what atoms we can see at this point. And what we should see would be the silver, the silicon on the surface. We should also see the iron oxide core. So we should see silica and iron oxide if we look at this point. And that's exactly what we saw. We saw some carbon, oxygen, and copper. Those are from the polymer coatings, from the background and from the grid below. But we do see silica and iron at this point, sp12. If we shift the focus of our TEM onto the tip here, we can focus the TEM down. We shine just on this part. We shouldn't see any iron because we're missing that. We're looking down here, we should see silica, but no iron. And again, that's exactly what we found. The sp10 region, we do some elemental analysis. There's no iron at that point. There is silica. So now we're pretty sure that this faint shape around the surface of our particles is a glass coating with an iron oxide core. We can work out from TM that it's about a 20 nanometer core, about a 2.6 nanometer glass coating. And that's where we attach the DNA primers. The gold particles are much bigger. So this is a gold particle. These are about 50 nanometers across. This is the particle diameter fitted to this normal distribution. We can see it's just below 50 nanometer average particles. If we put mercaptobenzoic acid onto the surface of these, we get a very strong SIRS signal. So this is from a SIRS laser. We shine a laser on, we can see this signal from each individual particle. So this is what we look for when we're looking to quantify the particles. The size of these peaks, we know how many particles we're looking at. If we do the PCR process, I get a sequence of DNA, I put it onto the PCR process with no nanoparticles involved, just simply PCR. We have a detection limit of about 10 femtomoles. So this is the PCR process going through. We can change the concentration of the DNA and see how it maps across. We can see the fluorescence increases. We can plot the CT value. We can plot that against concentration. And what we see is at low concentration, there's no coloration. It's fairly flat. It's a positive test, but it's fairly flat. And then suddenly you get a concentration dependence. You can start to quantify the amount of DNA. And if we add in some nanoparticles to this, just to see what happens, it doesn't affect the PCR process. If we do the same thing with our nanoparticles, so now rather than looking at fluorescence in cyber green, we're looking at the SIRS signal, and that SIRS signal should change with time, with concentration. So this is a picture of the SIRS signal. These are the two peaks we're looking at. And we can see if we decrease the DNA concentration, we decrease the peak size, we can still see peaks down here. This is now at 10 zemtomolar. So this is thousands of times more sensitive than the cybergreen method. And we get a very nice calibration curve. Unfortunately, the plot has gone wrong. The numbers aren't showing properly, but this is DNA concentration. This is the height of this peak increasing. And we can plot that out, a very nice straight line fit with concentration. So what we find is the SIRS system has a detection limit of 10 zemtomole. That represents about 6,000 copies of target DNA in the sample. It's possible that the real-time detection limit is well below this. In the normal PCR process, you get exponential growth. You go from 1 to 2 to 4 to 8. With this method, we're only extending one of the primers. So rather than getting exponential growth, what we'd expect to see would be linear growth of signal. So this is prediction, this is modeling. Normal PCR, you'd expect exponential growth with CT number. With this process, we'd expect to see a linear growth because we're only extending one primer. And then that's the modeling data. This is the real data in real life. This is the CT value of the intensity going up. And this is the linear increase. So this is really nice to see that this is the real data taken from our system showing the linear increase with PCR compared to the exponential increase. And that's exactly what we predicted at the start of the work. Finally, the DNA target, the cybergreen method, fluorescent method, has a detection limit of about 1 × 10⁻17 moles of target DNA, whereas the gold nanoparticle with the SIRS readout has a detection limit of about 1 × 10⁻20 moles of DNA. So the system has about 1,000 times more sensitive than the qPCR, which means we can do more detection. We can detect in a shorter time. We can detect different concentrations. And that brings me to the end of my talk, which looks pretty much on time, I think. So thank you for listening. I'm going to stop sharing and I'm going to open the floor for questions.
Thank you, Dr. Drake, for the fascinating explanation about the COVID testing and nanoparticle for enhanced limit detection for COVID. Okay. So is there any question for students? Anyone?
Silence. They're all completely busy.
Okay, so I will start.
I think your students are the same as my students. They're all very quiet.
Oh, is it the same? It's very new kid. Okay. Okay. There is one question from Tiara. Okay, please, Tiara, please. The floor is yours.
Okay. Good afternoon, Dr. Drake. Previously, I apologize if my question has been explained before because I didn't catch it well because it was raining in my house, so the signal was a little bit disturbed. I wanted to ask, based on my personal experience, when I had a COVID virus, at the time when I took the COVID-19 test through an antigen test, and the result was that it's positive. So that means the result, there's no antibodies in my body, and that also means that there was no COVID-19 virus in my body. But why, when I took the next test, the PCR test, the result was positive? Why did it happen? Should I believe the antigen one or the PCR one? So the result.
So you took an antigen test, a lateral flow test, and that came back negative.
Yes.
And then you felt ill, so you had a PCR test, and
yes.
the PCR test came back positive.
Yes. Why did it happen? I couldn't understand that. So why did it
happen? The PCR test is a lot more sensitive. So if I go back to let's go back to my talk. Where are we? The PowerPoint slides, share. If I go back to the timeline of infection, is it that one? Not that one. Too many slides. Where are we? All the way back to the beginning. Oh, there we go. So this is the slide of interest. When you're first infected, you have a high virus load in your saliva, in your sample. So the nucleic acid test works very well, but you have quite a low antibody level and quite a low level of proteins in the blood. So in your saliva, the protein system doesn't work very well. So when it's early infection, just a few days after you've been infected, when you start to feel ill, the PCR test is a lot more accurate and a lot more sensitive than the lateral flow test.
Oh. Okay.
Does that answer your question?
Yes. Thank you so much, Doctor.
I think it's quite an interesting point because at the moment, especially with Omicron, we're finding that people are testing negative on lateral flow in the morning, and then in the evening, by the evening comes along, they're starting to test positive. but they're already infectious. So they're going to events, they're going to football matches, especially football players, they're mixing and they're infecting other people and they didn't know they had it. So it seems Omicron is more infectious and that's not being picked up by the lateral flow tests as quickly. So to be negative for a lateral flow, you have to have 2 tests 2 days apart. So your first test on Monday could be negative. You've got to do a second test on Wednesday and self-isolate in between, then you know you're negative. Just one test doesn't really tell you anything anymore.
Thank you so much, doctor.
Okay.
Okay, is there any question? Okay, Timotheus, please.
Thank you. As we know now, there is many variants of COVID-19 from Alpha to Omicron. My question is, are these methods could work on any variants? And if it's so, is it necessary to change any chemicals? Thank you.
Yeah, so the tests are all based on the spike proteins. And the spike proteins are going to change with different variants. So the Delta variant, the Omicron variant have slightly different structures to the spike proteins. It doesn't seem to be affecting the PCR of a lateral flow. So the PCR is designed, the whole sequence of the virus genome is one big long sequence. And we know there's certain sections of that where mutations occur. We know there's certain sections which are very consistent and always the same. So the PCR method is designed to look for a certain section of the virus RNA, which we know hardly ever changes. So the PCR method should work for many different mutations. When you're detecting an antibody using the protein, it's possible that the mutated spike protein won't give a positive lateral flow test. We haven't seen that yet at all, so that still works very well. Equally, with vaccinations, you're vaccinating against a very particular sequence of the spike proteins. And we know now that for Omicron, the vaccination level has dropped quite a lot. But after 2 or 3 vaccinations, after the 3rd vaccination, your protection goes up. So your body does adapt. What you're doing when you get the vaccination is you're telling the body to look for this kind of protein. But your body also adapts and changes its approach. So we have a very intelligent immune system. It doesn't just look for one sequence, it changes itself as well. So our bodies are quite adaptive. So our immune system does adapt and does cope. So we still have quite good of protection against Omicron. It's possible that later mutations will change. So every year we might have to change the virus, change the sequences we're putting into the vaccinations. So we adapt our vaccinations as we go along. Hopefully, we can do that. Hopefully that will work fairly well. The new vaccination technology that's been developed for COVID is an amazing set of technology, all based on RNA. So we're not infecting, we're putting in the blueprints for our own bodies to produce the proteins to give ourselves antibodies. So that's very easy to change in the future. Fingers crossed.
Okay.
Sorry, that was quite a long answer.
Yeah, it's okay. Thank you, Dr. Greg. And there is one question from Faiza. If the blood test or lateral flowing test is less accurate than PCR, why we still use it? And is it true that the lateral flowing test can detect the dead virus? Is it?
Yes. So the PCR test is the best method. It's the most accurate. It's the most reliable. But the PCR test needs to have a trained operator. It needs to go to a lab. You need to have a very well-trained scientist running the PCR process and reading the data and giving you a result. The lateral flow test is very simple. You can do it at home. I do them every morning. You go down, you do a swab in your own mouth, you put a little drop onto the lateral flow system. Anyone can do it. So the lateral flow test is cheap and easy to run and can be run by lots of people, where the PCR test needs to be sent off to a lab. You need to have a trained operator. You need to have very expensive equipment to do that. So there's a trade-off between time and cost. And I've forgotten the second half of that question.
The second one, is it true that the lateral flow test can detect the dead virus?
Yes. Yeah, it is true. When the virus is in your body, you might not have enough to be infectious, but you might have a lot of dead viruses which your immune system has already killed off, and their proteins on their surface, the spike proteins, can still be in your saliva, even though you're not infected. And that could still give you a positive test on the lateral flow. Equally, the PCR can still give you positive, even when you're coming out of infection. You could still have some virus RNA inside your blood or inside your saliva, and that could still give you a positive test for a while after you recover.
So, like when we feel like secure, we are already like recovery from the COVID disease. Like how long is it? Like
typically we think infections last for about 2 weeks. So once you've tested positive, it should be about 2 weeks after that you can decide that you're now fit to go out. You could do more lateral flow, make sure the lateral flow comes down to negative, or you get a negative PCR test. But some people could still test positive several weeks after the infection's finished. So it's not particularly accurate. So it's about us knowing the virus and knowing what's infectious and what's not infectious. And that takes a lot of time.
Okay, thank you. Is there any question? We have five more minutes. Okay, there is a one question in the chat box from Miss Intan. It's a very nice presentation, Dr. Greg. I have two questions regarding the DNA testing. First, why does the surface of coronavirus only react with IgG? And the second one, which one is more accurate and fast for COVID testing using blood or saliva sample? Thank you.
So the surface coronavirus doesn't only react with IgG. The virus produces its spike proteins as a way to get into cells. So the virus uses, it's like a key. Imagine you're walking down a street and you've got a few keys. You want to find the right key that opens the door to get into a house. And coronavirus tries to do that by using its spike proteins. Those are the keys to get into cells. Our IgG is what our body develops to block those keys. So we develop IgG to stop coronavirus from doing that by blocking their keys. So we make IgG to recognize the spike proteins. And that's how the vaccinations work. We expose our bodies to some of the spike proteins, not the virus itself, just the spike proteins, which by themselves are harmless. They go into our body recognizes them and develop IgG to bind to those spike proteins. So then we can block the virus when it comes. In terms of testing, I would say that the blood is probably a better one to use, but saliva is much simpler. So you don't want to go around doing a pinprick on your finger every day because it's quite painful. Whereas doing a saliva sample is very quick and easy. The problem with saliva is that it's affected by what else you've had in your mouth. So if you've just eaten an orange, for example, the saliva in your mouth will be very different. It has a quite acidic environment. So your saliva isn't always the same. It depends on what you've been doing beforehand. So ideally, you would always do this test first thing in the morning. You wake up, you've been in bed all night, you haven't had much to eat. So your saliva is fairly constant, and that would be a good time to test. Once you've eaten some food, you've brushed your teeth, the saliva will change in your mouth. So it's not always accurate. Which is why you also do the nose swab, which is slightly more accurate. But even noses, we're inhaling dust all the time. So our nose swabs are also variable in quality, whereas blood is always the same. But blood is painful to get, so saliva is easier.
Okay, thank you. I think one more question before we close the session. Is there any question? Okay, I think
All finished.
yeah.
So if you do have any
questions.
questions, feel free to email me or Twitter me or whatever you like. I'm always happy to contact people.
Okay, thank you very much. Thank you very much, Dr. Greg. So we are in the end of the session. Thank you very much for your time and your talk. That is very fantastic. Okay, thank you again, Dr. Greg. I hope you have a good day. Have a good day. And, yeah.
All my Christmas holidays now, so bye-bye.
Bye-bye.
Christmas time.